Welcome back to my channel. Today we're going to do the high yield revision of chapter genetics for MRCOG part 1. This is the video of the continuation of the series of revision or the short notes. So let's start with the clinical application of the genetics, that is chromosomal aberrations. And the chromosomal aberrations, they can either be numerical or can be structural. So in the numerical, there is the aneuploidy, such that either there is a hyperploidy, which means that there is an increase in the number of the chromosome, and the other is the hypoploidy, similar that there is a decrease in the number of the chromosomes. Numerical means here the number defect. So what is the aneuploidy of the sex chromosome? The aneuploidy of the sex chromosome will be the Klinefelter or the Turners, and aneuploidy of the autosomal chromosomes includes the Down, Edward, and Patau syndromes. The other problem is non-disjunction. It is the pairs of how this numerical aberration happens. It happens either because of the one of the most important causes, the non-disjunction, such that they are the pairs of the homologous chromosomes at the meiosis 1 and the sister chromatids during the mitosis anaphase, such that when there are the two most important points when the non-disjunction occurs and lead to the aneuploidy. Number one is a pair of the homologous chromosomes at the meiosis 1, and the second is the stage when they are the sister chromatids during the mitosis stage. The most common aneuploidy is the Down syndrome, and the Down syndrome, it increases with the maternal age when the mother age is more than 30 years, and these aging leads to the aging of the oocytes, and 80% of the spontaneous pregnancy, they lead to the miscarriage. It is also called as a trisomy 21 because this trisomy occurs at the chromosome number 21. The prevalence is 1 in every 700 females, and the causes includes the mosaicism or the Robertsonian translocation at 1421 chromosome, and the primary trisomy, which and the most important or the major cause is the 90% is the non-disjunction as the meiosis. The most important clinical feature includes the general features and the craniofacial facial features. The general features include the mental retardation and the hypotonia and the conducting hair loss. The craniofacial includes the flat occiput, the brachiocephaly and the epicanthal fold, and the speckled iris, protruding tongue, and the prominent malformed ears and the flat nasal bridge. The most important cardiovascular problems are the atrioventricular canal or septal defects, and GIT includes the duodenal atresia and the Hirschsprung's disease. Down syndrome is the common of all because there is a less gene on the chromosome number 21, which means that there are the more chances of the survival of the Down syndrome. This is the most important example of the aneuploidy. The other example is the Edward syndrome and the Patau syndrome. So let's see Edward syndrome. What happened in the Edward syndrome, that it is also called as a trisomy number 18, and E for Edward and E for 18, and the prevalence is 1 in 300, and it has a higher mortality rate. By 1 month 30% of the babies they die, by 2 years 50%, and by 1 year 90% of the babies they died. The clinical features, the most important are the restricted growth and the defects, which are the overlapping fingers and the rocker bottom feet and the cleft lip and the palate, and the cardiac defect AST, VST, and PDA, and GIT defects includes the exophthalmos and the inguinal hernia. And the next important syndrome is the Patau syndrome, and this is also called as the trisomy 13. P is for puberty, and 13, mostly the puberty occurs at the 13, and it has the highest mortality rate among of all of the trisomies, and 100% mortality in the first month of the birth. And then what are the other symptoms, which includes that it has a weak muscle tone and the very small poor developed eyes and the extra fingers of the toes and the hypotonia and cleft lip and palate and scalp defect and the omphalocele. Klinefelter and Turner syndromes, they are the sex chromosome aneuploidy. The Klinefelter syndromes is the most common karyotyping findings that they are the 47 double XY and incidence is 1 in 1000 live births. Similarly, Klinefelter is also the common of all the other. Whenever there is a SBA which comes that there is the infantile tall men and who have small testes and the hypogonadotrophin and the hypogonadism, then your answer should be the Klinefelter syndrome. And then the another important aneuploidy sex chromosomal aneuploidy includes the Turner syndrome, and it is the monosomy such that X with 45X not or only 45X, and the prevalence is 1 in 2500, and they are intellectually normal females. The clinical features includes the streak gonads with the normal uterus and widely spaced nipple, no menstruation, deformed elbows and coaction of the aorta, and broad chest and widely spaced nipples, streak ovaries, and cubitus vulgus and coaction of the aorta are the most common clinical features by which these patients presents. Now, as we have done with the numerical chromosomal abnormalities, now let's see the most important structural abnormalities of the chromosomes. They occurs either because of the deletion, duplication, translocation, or inversion. The deletion and duplication, their amount of the genetic information in the chromosome, they can change. Translocation and inversion, the genetic material there remains the same, but it is rearrange. Then the reciprocal translocation, it is the most common reciprocal translocation in the human is T1122 and Q23 Q21 in the values chromosome or 11 and 22. It involves the two chromosomes, one break in each chromosome. The two chromosomes they exchange the broken segment. Robertsonian translocation is a chromosomal rearrangement that involves the fusion of the long arm of the two acrocentric chromosomes and loss of their short arms. By definition, Robertsonian translocation only involves the five acrocentric chromosomes 13, 14, 15, 21, 22, and most common Robertsonian translocations are ROB13Q, 14Q, and ROB14Q and 21Q. Then the deletion. How the deletion? In the deletion, the most important examples are the Cry-Duchat syndrome and 2022 and George cardiofacial disease. But other two important syndromes are the Angelman syndromes and the Prader-Willi syndrome. The Angelman syndrome, it is characterized by the 15 Q11 13 deletion, and in it is maternally derived chromosomes, whereas in the Prader-Willi it is again in the 15 Q11 13 deletion, but it is paternally derived chromosome and 5% associated with the paternal UPD15, and here in 25% cases no deletion but maternal UPD15. And then learning disabilities, both have the learning disabilities, and seizures and paroxysmal inappropriate laughter, macroglossia, and abnormal EEG. Whereas in the Prader-Willi syndrome, it is characterized by the 15q11 13 deletion in the paternally derived chromosome. In 25% cases, no deletion but maternal UPD15, and it leads to the learning disabilities and unital hypotonia with the poor suckling and hypogonadism, flat face, and almond shape, papillae vision, and small hands and feet. Then let's do some prenatal genetics because these are also again important. And what say we here that the prenatal genetics, it is very important branch of the genetics, and it has both invasive method and non-invasive method, and also the screening tests. So first discuss the invasive method. Invasive methods are the amniocentesis, which should be done more than 15 weeks of the gestation, and CVS, it can be done before 15 weeks, and cardiosynthesis. cardiosynthesis. Non-invasive includes the ultrasonography and maternal screening in first and second trimester and fetal DNA in the maternal circulation. Now the screening test and the diagnostics test. What are the screening tests? The knuckle transfluency, it is the most important screening test, and it is the measurement of the subcutaneous fluid between 11 to 14 weeks for the trisomy 21, 18 and 13. Then combined test. The combined test includes the knuckle transfluency scan and the true biochemical markers, which includes the PAP-A and beta-HCG. Then there is a triple test, which includes the alpha-fetoprotein, beta-HCG and the estriol. Alpha fetoproteins, they are decreased in the Down syndrome, important, as they decrease, they only decrease in the Down syndrome and trisomy 18, and it increase in the congenital malformations, for example omphalocele, bladder exotrophy, intestinal atresia and spinal defects as well. Now the quadruple test includes alpha fetoprotein, beta HCG and estriol and inhibin, and cell-free fetal DNA. It is used to detect the gender, rhesus T, blood typing, and to detect the trisomy 13, 18 and 21. Okay. Now the diagnostic test, they includes the amniocentesis, the CVS and the choriosynthesis. Amniocentesis, it is mostly done after 15 weeks. If done less than 14 weeks, there is increased risk of the fetal loss, and they're the most commonly used. Then CVS, they do not perform less than 11 weeks, never perform less than 11 weeks, and it includes aspiration of the placental tissue. And the choriosynthesis is again done more than 14 weeks of the gestation and done in the umbilical vein, and as it has the 1 to 2% of the risk of the miscarriages. Okay, then these are the important serum markers. The serum markers, and we see how these are changes in the triple test and quadruple test in the combined test for what we will see in the Down syndrome, adverse syndrome, and PA pregnancy associated plasma protein A. It is reduced in the Down syndrome as well as in the adverse syndrome. And then alpha fetoproteins, we know that we have studied that they are reduced in the Down syndrome and adverse syndrome. However, they are elevated in the neural tube defects such as the spina bifida and multiple pregnancies. And then unconjugated estriol, it is again reduced in both of the cases of the Down syndrome and the adverse. Inhibin A is elevated. However, inhibin A is elevated in the Down syndrome and it is reduced in the adverse syndrome. Again, beta HCG is elevated in the Down syndrome and reduced in the adverse syndrome. So let's move on, and here let's discuss a few important slides from the cell cycle. And we know there are the 20,000 genes in the human genome, and it contains of the nucleotide. The nucleotide, it is made up of the nitrogenous base, a phosphate and a pentose sugar. The pentose sugar and nitrogenous base together are called as the nucleoside, and the purines are the guanine and adenine, and pyrimidines includes the cytosine, thymine and uracil. And the chromosomes, they are mostly detected at the metaphase, and they're identified by the gene sustaining, and there are the 46 chromosome in the humans, 22 pairs of the autosome and 1 pair of the sex chromosome. And then the important the blots, and there are the northern blot. It is called as the northern blot. Here there is R and R. It's like R and R. Northern blot is for RNA, and southern blot, as it is opposite to north, it is the DNA, and then the western blot left is the western blot, which is for the protein. And then another trick we can apply over here is that how to remember the phases of the cell cycle. If we remember mnemonic of the PMAT, these are the phases of the cell cycle. And then what is the interphase? Interphase is the most busy phase of the DNA cell cycle replication. It has G1 phase, which is known as resting phase, and S, which is the cell replication, and G2, which is the spindle production. And what is the mnemonic PMAT? PMAT is for the prophase, M for metaphase, A for anaphase, and T for telophase. So what happened in the meiosis 1? There's a duplication and recombination in meiosis 1, and what is happens in meiosis 2? There's separations of the chromosome, and here you can see that there is this whole cycle is the this whole is the interphase. There, it includes the G1 phase, which includes the cell's maturation, S phase is DNA synthesis, and G2 phase, which is the cell preparing for the division. And then we come with the mnemonic, which is the PMMAT, PMAT, and then at the end is the cytokinesis. Okay, then let's see most important types of the RNA. There is a messenger RNA. The messenger RNA, it carries the coding of the instructions of an amino acids, and their sequence of a protein is the transcription. Then transverse RNA carries the specific amino acids to the ribosomes to form the polypeptide chain translocation, and then rRNA, which is the ribosomal RNA. So the two important things: what is the transcription? What is the translation? The transcription, it includes the synthesis of DNA from a synthesis of RNA from DNA, and then translation is synthesis of protein and RNA. Okay, how you are going to memorize this? That C comes first in the transcription. The C comes first. Okay, just remember the C comes first, and then L comes later. Transcription and translation. We know that the transcription will come first and translation will become later. And then how you are going to remember next thing, that we have a DNA, and DNA needs the replication. What happens is these two strands, they are going to form the RNA first. So the transcription is a process or the synthesis of RNA from DNA, and then these chromosomes, these RNAs, they are going to be translated. Like there will be some protein which is going to translate this RNA and will convert it into a specific protein as an end product. So you understand this, how you're going to memorize the transcription and translation by first we need RNA in order to form a protein. So transcription will occur first, and that transcription, that mRNA is then translated into a protein. Okay, so these are the most important points, and here we are going to conclude our today's session. Hope you like the video and hope you understand all things. Until next time, take care.